In this video, we want to talk about an aspect of DNA structure that's beyond the double helix. And I don't really have any notes for it in your study guide. So I just want you to take a blank sheet of paper out, and it shouldn't take too long. The topic has to do with the idea of DNA compaction. And what that is how do we take DNA? Here's this beautiful structure of double-stranded DNA. We call it B-form nucleic acid helix. You can see the major groove out front here, the minor groove here, everything we talked about previously. How do we take that and get it inside a cell? If you took the largest chromosome in your body, chromosome number 1, and stretch it out, like this structure right here, it'd be 7 centimeters long. It's too big. Also, what about the structure of DNA that we're used to thinking about in cells? What about the chromosome? How is it that these two things are the same thing? DNA and chromosomes, they both contain DNA and the genes that we have in our body. Well, it has to do with compaction. That is, how do we take a structure and compact it down into a small space? And the answer is we do what's called the creation of a nucleosome. And a nucleosome is going to have a core protein, kind of looks like a tuna can here. And this is the histone protein. It's called a heterooctamer. Do you remember that terminology we used back in our protein structure? Octamer, 8 subunits, hetero meaning they're not all the same. The histone protein is this red tuna can shaped object we have here, and it is covered with positive charges. If I asked you to think about what amino acids might we use to have positive charges on a protein, I hope you'd say things like histidine and lysine and arginine. In fact, lysines make up 25% of this molecule. It's a huge, big ball of positive charge. That's the core of the nucleosome, but it's really about the DNA that we're interested in. And the DNA I'm going to show in green here. And the DNA is about 150 base pairs. The green line I have shown there, wrapped around there, is actually double-stranded DNA. And it's wrapped twice. Now remember, DNA is highly negatively charged, and the negative charge of the DNA is attracted to the positive charge and helps it wrap right around there, like you might wrap a wire around a tin can or something like that. So you end up with this structure, and this is wrapped, and we call this wrapping of the DNA supercoiling. And what it is we have coils, of course, helices with the DNA wrapped around itself, but then we take those helices and make another overhelix of it. And what this ends up looking like, we're going to show you that on the next page here. This is not, again, from your notes, it's just a figure from a textbook. And what we have here is the nucleosomes right here, the histone, DNA wrapped around it, and you end up with a kind of a thing that's like pearls on a string, where the nucleosomes are the pearls and the whole DNA molecule is the string. These nucleosomes now stick to each other, and they actually form helices of helices, supercoiling. And those helices or helices form helices, and then they form helices, and then they form helices. And it's kind of like when you take a rubber band and twist it and twist it and twist it, eventually it forms really compact, almost like knots. And eventually you do this 6 or 7 layers of helix, and you end up with what we are typically seeing in cell biology, which is a compact, solid, very stable metaphase chromosome.